Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we begin Chapter 13, Entrepreneurship. Biotechnology entrepreneurship turns a scientific opportunity into a product or service that solves a real problem responsibly. That journey is much more than starting a company or presenting an impressive idea. It requires evidence that the science works, understanding of users and markets, a capable team, facilities, and biological resources, stage-appropriate finance, reproducible manufacturing, quality systems, biosafety, regulatory planning, intellectual property awareness, and public trust. By the end of this lesson, you will be able to convert an observation into a clear problem-opportunity statement, explain entrepreneurial qualities, compare entrepreneur and manager roles, build an enterprise plan, map resources and funding, trace a biotechnology venture through evidence milestones, choose suitable intellectual property strategies, and explain how consent, provenance, and benefit sharing protect against biopiracy. Our roadmap begins by distinguishing a scientific observation from an entrepreneurial opportunity. We then study curiosity, initiative, resilience, ethical judgment, communication, teamwork, and calculated risk-taking. We compare the entrepreneur's role in creating and bearing uncertainty with the manager's role in coordinating defined operations, while recognizing that one person may perform both. Next, we build an enterprise plan around the customer problem, value proposition, scientific evidence, market, competitors, operations, milestones, risks, and finances. We map technical expertise, biological materials, facilities, data, manufacturing and partnerships, then match grants, loans, angels, venture capital, and revenue to different stages. Biotechnology-specific sections cover long development cycles, variability, quality, biosafety, approval, and public trust. Finally, we examine patents, copyright, trademarks, designs, confidential know-how, freedom to operate, biopiracy, informed permission, provenance, and equitable benefit sharing. Entrepreneurship recognizes an opportunity, organizes resources, creates value, and accepts managed uncertainty. Begin with a problem statement, not a technology name. Identify who experiences the problem, where and how often it occurs, what solution is currently used, and why that solution is inadequate. Then connect a biotechnology observation to a possible improvement. For example, an enzyme that detects a contaminant rapidly is scientifically interesting. It becomes an opportunity only if users need faster detection, the result is sufficiently accurate in real samples, the device can be manufactured, and the cost fits the setting. Interview evidence, workflow observation, and willingness to adopt test the need. Laboratory controls test the mechanism. Keep these evidence types separate. A useful opportunity statement links a specific user problem, proposed biological solution, measurable benefit, evidence already available, critical assumption, and next validation step. Entrepreneurial qualities are behaviors used to make decisions under uncertainty. Curiosity asks why a problem persists and which assumption should be tested. Initiative converts that question into a small ethical experiment. Creativity proposes alternatives, while technical competence evaluates biological feasibility. Calculated risk-taking does not mean ignoring danger. It compares expected benefit, probability of failure, consequence, and possible mitigation. Resilience means learning from a failed experiment or rejected proposal without hiding evidence or repeating the same plan blindly. Communication helps scientists, users, regulators, investors, and partners understand one another. Teamwork combines expertise that no individual possesses alone. Ethical judgment sets boundaries when commercial pressure conflicts with safety, consent, or truthful reporting. Confidence is useful for action, but it never replaces data, market validation, quality, or regulatory responsibility. A strong exam answer connects each quality to a concrete decision rather than presenting adjectives as a memorized list. An entrepreneur commonly originates a venture, assembles initial resources, and accepts uncertainty about the problem, solution, and business model. A manager coordinates resources toward defined objectives through planning, organization, staffing, monitoring, and correction. This is a functional distinction, not a ranking of personal importance. During discovery, entrepreneurial work dominates because assumptions about need, science and delivery are still being tested. As the venture develops, managerial systems become essential for quality, schedules, budgets, documentation, and reliable production. A founder may perform both roles initially, then recruit specialists as complexity grows. Too much exploration without management creates missed milestones and inconsistent evidence. Too much routine management before the opportunity is validated can efficiently execute the wrong plan. A good comparison uses the same basis on both sides: primary focus, type of uncertainty, decisions, time horizon, resource responsibility, and success measures. Mature biotechnology ventures continually combine exploration with disciplined execution. An enterprise plan is a connected set of hypotheses, evidence, activities, and financial consequences. It defines the user problem and value proposition, describes the product or service, summarizes scientific evidence, estimates the reachable market, and compares alternatives. It explains how the venture will operate: facilities, suppliers, biological materials, data, manufacturing, quality control, distribution, and support. Biotechnology plans must include research uncertainty, scale-up, biosafety, ethics, and approval dependencies. Milestones should be evidence-based, for example, achieve a validated detection range in real samples, or demonstrate a reproducible batch specification, rather than vague tasks such as continue research. Each milestone has cost, time, responsible team members, failure criteria, and a decision to proceed, revise, partner, or stop. Financial projections should follow operational assumptions and acknowledge uncertainty. A plan is not a prediction guaranteed to occur. It is a disciplined model updated when evidence changes. A biotechnology venture needs more than a brilliant scientific founder. Build a capability map from present evidence to product delivery. Scientific skills may include molecular biology, microbiology, cell culture, bioinformatics, or process engineering. Clinical or field expertise ensures that experiments represent the intended setting. Quality and regulatory specialists translate requirements into documentation and controls. Commercial skills cover user research, partnerships, pricing, and distribution. Physical resources include laboratories, instruments, storage, containment, reference materials, and reliable suppliers. Samples and biological materials require permission, traceability, and suitable handling. Data systems protect integrity, privacy, and analysis. Scale-up may require a contract manufacturer or technology partner rather than immediate ownership of a plant. For each milestone, list the capability required, currently available resource, gap, method of access, cost, and lead time. Partnerships reduce some capital needs, but add coordination, intellectual property, and quality responsibilities. Funding sources differ in repayment, ownership, control, timing, and evidence expectations. Founders and family may finance early exploration but carry personal risk. Grants and incubators can support research, infrastructure, or mentoring without taking ownership, though they have defined purposes and reporting obligations. Loans require repayment and are usually better suited when cash flow or assets make repayment credible. Angel investors provide capital and experience in exchange for ownership. Venture capital commonly seeks high growth and future returns, requiring strong evidence, a scalable market, and governance rights. Strategic partners may provide facilities, distribution, or technical capability, but can create dependency or exclusivity. Revenue is powerful because it validates willingness to pay, yet many regulated biotechnology products cannot generate revenue during early development. Match finance to the next value-creating milestone and fund enough time to reach it. Understand dilution, repayment, control restrictions, and failure consequences before accepting money. Biotechnology venture development often differs from a simple digital service because living systems are variable and products interact with health, agriculture, or ecosystems. Cells change with passage and environment. Microbial production depends on strain, stability, media, oxygen transfer, and process conditions. Biological samples differ between individuals and locations. Scale changes mixing, heat transfer, and contamination risk. Specialized facilities and trained personnel increase cost and lead time. Clinical, food, agricultural, or environmental applications may require formal safety and performance evidence before sale. Manufacturing must reproduce identity, purity, potency, or other critical quality attributes across batches. Cold chains and shelf life can affect distribution. Public trust depends on transparent claims and responsible engagement. Software can sometimes be updated rapidly after release. A biological product or released organism may be difficult to recall. Therefore, biotechnology entrepreneurship joins scientific innovation with quality by design, risk management, regulatory strategy, manufacturing, and long-term monitoring. A disciplined biotechnology startup pathway uses stage gates. First, confirm the unmet need and intended user. Next, establish the scientific principle with appropriate controls and define a prototype. Examine ownership, patentability, and freedom to operate early enough to influence design. Map biosafety, ethical, and regulatory requirements before expensive development. Validate the prototype in increasingly realistic samples or models, and define performance specifications. Build the team, business model, and financing plan around the next evidence milestone. Pilot manufacturing tests, reproducibility, contamination control, supply and quality. Field or clinical validation tests, performance in the intended context. Market evidence assesses adoption, payment, and distribution. At each gate, compare results with predefined criteria and decide to proceed, revise, partner, license, or stop. Iteration is normal. Evidence may return the venture to an earlier stage. Stage gates prevent enthusiasm from committing unlimited resources to an unvalidated assumption. A prototype demonstrates a function. Production must deliver that function consistently. Define critical quality attributes such as identity, purity, potency, concentration, sterility, sensitivity, or stability according to the product. Identify process parameters and material properties that influence those attributes. Raw materials and biological inputs need specifications, qualified suppliers, and traceability. Standard procedures, calibrated equipment, training, and documentation reduce avoidable variation. In-process controls reveal problems before the final batch. Release tests decide whether a batch can be used, but testing a few final samples cannot correct a poorly controlled process. Stability studies establish storage conditions and shelf life, while packaging protects the product during transport and use. Deviations require investigation and corrective action. Scale-up should compare the larger process with the validated smaller process and assess new mixing, transfer, or contamination risks. Quality is scientific evidence converted into repeatable operations. Intellectual property systems protect different forms of creation under different conditions. A patent may protect an invention that meets legal requirements such as novelty, inventive step, and sufficient disclosure. Protection is territorial, time-limited, and not automatic permission to commercialize. Copyright protects original expressions such as manuals, software, or illustrations, not the underlying scientific fact. Trademarks identify the commercial source of goods or services. Design protection concerns qualifying visual appearance. Trade secrets may protect confidential protocols, formulations, or know-how, while reasonable secrecy measures are maintained, but secrecy may be lost through disclosure or independent discovery. A biotechnology venture may combine several strategies, for example, patenting a process, keeping manufacturing optimization confidential, registering a brand, and using copyright for training material. Publication, collaboration, and employment agreements affect ownership and timing. Competent professional assessment is important because exact legal requirements vary by jurisdiction. Patentability and freedom to operate answer different questions. Patentability asks whether an invention may qualify for protection in view of earlier public knowledge. Freedom to operate asks whether making, using, or selling a product in a particular territory may fall within valid claims owned by others. A company can obtain a patent on an improvement, yet still require a license to a broader underlying invention. Conversely, a useful product may be commercialized without its own patent if no blocking rights apply, though competitors may copy it. Review inventorship and ownership, especially when work involves universities, employers, funders, or partners. Map relevant claims, territories, filing and expiry dates, licenses and confidential obligations. An online search is a starting point, not a legal conclusion. Intellectual property strategy should support the business model and responsible access. It should not replace scientific validation, quality, or customer value. Biopiracy concerns unauthorized or inequitable appropriation of biological resources or associated traditional knowledge. A plant, microorganism, or community practice may guide a valuable product, but commercial development does not erase the rights and contributions connected to its origin. Responsible practice documents provenance, where material or knowledge came from, who provided it, what permission was granted, and for which use. It follows applicable access and benefit sharing requirements and obtains prior informed consent when required. Mutually agreed terms can address monetary and non-monetary benefits such as royalties, research collaboration, training, infrastructure, data access, or shared ownership. Confidential or sacred knowledge needs appropriate protection. Later, genetic sequencing or laboratory modification does not automatically cancel earlier obligations. A strong answer distinguishes lawful discovery from equitable use and explains that attribution alone may not satisfy consent or benefit sharing. Transparency and continuing partnership build trust and reduce exploitation. A biotechnology enterprise manages several connected risks. Technical risk asks whether the mechanism and manufacturing remain reliable. Biosafety examines exposure of workers, users, communities, and ecosystems. Regulatory risk arises when evidence or processes do not meet applicable requirements. Financial risk includes delays, cost growth, and unsuitable funding. Ethical and social risk includes invalid consent, privacy loss, inequitable access, misleading claims, or community exclusion. Build a risk register with probability, consequence, early warning indicator, mitigation, owner and review date. Containment, quality controls, secure data, consent procedures, and independent oversight should be designed before failure occurs. Communicate evidence at the correct level. A prototype result is not a proven public benefit. Report limitations and negative findings, and correct claims when evidence changes. Trust grows from competence, transparency, and accountability, not promotional confidence. Responsible conduct is part of enterprise value, because unsafe or deceptive growth is not sustainable entrepreneurship. For a CBSE answer, define entrepreneurship as recognizing opportunity, organizing resources, creating value, and accepting managed uncertainty. Convert a biotechnology observation into a problem statement by naming user, setting, current alternative, proposed biological solution, measurable benefit, and next evidence. When listing qualities, connect each quality to a decision. Compare entrepreneur and manager using the same basis and state that one person may perform both roles. Draw enterprise planning as problem, value, evidence, market, operations, team, milestones, risks, and finance. Match funding sources to discovery, prototype, validation, and scale up, noting repayment, ownership, and control. Explain why biotechnology needs specialized facilities, long evidence cycles, quality, biosafety, and regulation. Draw a stage-gated startup pathway. Distinguish patentability from freedom to operate, and patents from copyright, trademarks, designs, and trade secrets. Define biopiracy and end with provenance, permission, benefit sharing, transparency, and limitation. Close your notes and rebuild the chapter. Entrepreneurship recognizes opportunity, organizes resources, and creates value under managed uncertainty. A discovery becomes an opportunity only when it solves a validated user problem through feasible delivery. Curiosity, initiative, resilience, calculated risk-taking, communication, teamwork, and ethics guide action, but confidence never replaces evidence. Entrepreneurs explore opportunity while managers coordinate defined operations. Ventures need both. An enterprise plan joins value proposition, science, market, operations, team, milestones, risks, and finance. Capability maps reveal resource gaps. Funding sources differ in repayment, ownership, control, and stage fit. Biotechnology adds biological variability, specialized infrastructure, quality, biosafety, regulation, and public trust. Stage gates move from need and proof of principle through prototype, production, and market validation. Patents, copyright, trademarks, designs and trade secrets protect different assets, while freedom to operate remains separate. Responsible biodiversity use requires provenance, permission, recognition, and equitable benefit sharing.